Hi,
Oh my gosh, I am dying.
what's wrong?
I'm trying to like make a Google Doc based on like what Merck's, I don't know how to say it, uh, based on what she said. So I still need to like work on this.
Okay.
The issue is like I haven't really worked on the probability problems, so I still need to like work on that, or like I need to understand it.
What is that even about?
Uh, like the sum and product rule thingy?
Oh, yeah?
Yeah, I haven't really looked at those problems.
I'm gonna add a song to your playlist.
One, hold on. I was studying.
I haven't really studied, I'm just trying to finish the quiz that I made.
Oh my gosh, thank gosh. Not a lot of people know have studied it. Yeah. Which is I'm kind of scared for.
Well, it might be really easy.
That's true. Actually, I don't know, because like I'm scared that he's gonna ask us like really confusing questions where it's like, oh, is it like which is which is the answer, A and B or all of the above? I'm like scared of those questions.
Oh, yeah, those questions make me second guess myself.
Doo-doo.
After this class, I need to study for chemistry.
Oh my goodness, me too. I'm scared of that. Is that like all of yours? All of your finals?
Yeah, just this class and then chemistry, and I'm done.
I see.
How about you?
I have chemistry and what else do I have? Oh yeah, I have physiology.
Oh, good luck on that.
Thank girl. I don't think it's gonna be that bad. I mean, it might be, but I'll be fine. I've done the lab. I expect it to not be that bad. Maybe. I'm gonna go BRB so then I can like continue writing this down.
Okay. Hello.
Hello.
Oh,
Hello.
anything you guys want to study?
Are these your slides?
No, this is just my Quizlet. I'm just
Oh yeah, that's what I
adding
meant.
to
My
it.
bad. Do you have a link to this?
Yeah. Mm, let me just. Oh, whoops. Why did I do that? Reflex. Mhm. Okay.
Thank you.
Mhm.
Oops.
Just okay.
How are you feeling about the final?
I don't know. I haven't really studied, but I
Uh,
think if I look at the paper, I'll get it.
I believe in you.
Thank you. You guys want to study anything?
I think like focusing on any of the stuff that the person said would be nice. I don't know if
Okay.
the others would agree.
Oh no, they're kind of offline.
Oh,
Let's see. What did they talk about?
let's see.
Transcription and translation. Okay. Hmm. I forget what lecture. Oh, it was a while back, maybe? No. What I want to see. Hmm,
Well, I know translation is 22,
22.
but I'm not sure about the other one.
Let's see. I think they might be in the same.
Yeah, that's what I was thinking.
Or was transcription earlier? I got too many tabs. Mhm. Is it right? Sketch plan. Mm. Mhm. Mm. Transcription might be with like promoters. Mm. Okay, whatever. Let's talk about translation. Okay. So translation is taking mRNA and turning it into an amino acid sequence. And this happens in the ribosome. Okay. And the ribosome has a large subunit and a small subunit. And then, like when translation is finished, the two subunits separate, and then they go find other subunits to partner up, and then do translation with. Yeah, this is like the central dogma of molecular biology. So you start with DNA that is made in the nucleus, and then you go through transcription, and then the DNA is made into messenger RNA, and then the messenger RNA is brought to the ribosomes where it is translated into a amino acid sequence, and then that sequence makes proteins. Yeah, and then these are like the building blocks. So DNA is GATC, RNA is GAUC, and then their DNA and RNA both made in the 5' to 3' direction. Or like they're red that way. And then protein, you have the N-terminus to the C-terminus. And then the coding problem was like, how does DNA become protein? Like, how do these individual nucleotides become amino acids? So basically there's like 20 amino acids that code for or yeah, there's 20 amino acids that like make up proteins. And then DNA code is overlapping, I think it was. Like, one amino acid might have like 3 combinations. Like the stop codon, there's like three codes for the stop codon. And then this experiment was just showing that like the DNA code is a triplet code. Like amino acids are grouped in triplets. Wait, no. So nucleotides are grouped in triplets, and then each triplet makes an amino acid. Yeah, these three guys, or these 4 guys, they basically mm, they took like a bacteriophage and then like inserted bases into like the okay, I don't remember. Yeah, I need to go look at my notes for this. Okay, so they used bacteriophages and then they inserted nucleotides into the viral genome of the bacteriophages. Yeah, and then when they inserted 1 or 2 nucleotide bases, the protein that was created with the viral genome was not functional. And then when they inserted 3, the protein was still working. So the experiment shows that the DNA code is non-overlapping. Wait, I said overlapping earlier, but it's non-overlapping. And then, so basically insertion of 1 to 2 nucleotides messes up the reading frame, but inserting 3 nucleotides would restore the original amino acid chain. So the length of the DNA code is 3 nucleotides. And then this is the RNA Tide Club. They basically got together to discuss like how the nucleotide bases become amino acids, but they couldn't figure that out. So then these 2 guys, they basically figured out like they created like a codon chart for like each and every combination of nucleotides and like what those combinations of nucleotides would make. Yeah, they basically took quite a while to finish this because it was a lot of work. And then there's like extra codons because there's 20 amino acids and then there's like 64 different combinations or something like that. So there's like extra codons, and those extras would just be, they would just make like the same amino acid. So like one amino acid would have like 3 combinations. And then the genetic code also has the start and stop codons. The start codon is AUG, which is methionine. And then stop codon is like UGA, UAA, UAG. And then basically on the exam, he's just gonna give us like a strand of DNA code, and then we gotta translate it using the codon chart. And then how we start translating is we look for like where methionine is, and then we start there. So AUG is here. Then we just start reading here. And then all the way until the stop codon. Oh yeah, this is the N-terminus. This is C-terminus. And that's all we need to really know about like how proteins are. We don't need to know like amino groups or carboxyl groups. Yeah, peptide chains are just a bunch of amino acids connected together. Oh yeah, there's different structures. So primary structure is just amino acid chain, and then the secondary is alpha helices and beta pleated sheets. Just like the chains are like folded in a certain way, and then they become alpha helices and beta pleated sheets. And then tertiary is just like is folded even more, and yeah, it's folded even more, and then there's like sulfide bonds in between like the chains. And then for quaternary is just like a bunch of tertiary in a like connected together or like attached to each other. And then the central unit of translation is the ribosome. Yeah, and then these people discovered like ribosomes, what it does, and its structure. And then we have 3 types of RNA: messenger RNA, which makes amino acid sequences, and then ribosomal RNA, which organizes protein in ribosomes and catalyzes protein synthesis. So the ribosome is mostly made of RNA, and then it also has protein, but it's mostly made of RNA. And then transfer RNA shuttles amino acids for protein synthesis. It's this stamp looking thing. So the top is where it carries the amino acid, and then this bottom part is it has an anticodon, which is complementary to the mRNA that is like put into the ribosome to be read. So yeah, and then like if the anticodon matches the mRNA that is read, then amino acid is placed down, and then the tRNA leaves to get more amino acids, and then there's more tRNAs like coming around in a circle, and then they leave. They like put down amino acids and then they leave. Yeah, like this. Now this is a large subunit, small subunit. Okay, so there's like 3 phases to translation. Initiation, the ribosome assembles itself. the large and small subunits come together, and the first tRNA attaches to a start codon, which is methionine. And then elongation is where the tRNA places its amino acid, and then the ribosome moves to the next codon. So like amino acid is placed down, ribosome shifts that way. Yeah. And then the next tRNA comes in and places the amino acid. And then termination is when a stop codon is like placed down by a tRNA, and then the ribosome breaks apart, and then the protein strand is free. Yeah, and then the protein just goes to the Golgi where it's packaged, and then the rough ER also does packaging and cleaning up. And then oh wait, no, it starts from the rough ER and then goes to the Golgi. Golgi just like packages stuff and then sends it to like the vesicles, little vehicles that go outside of the cell. And then that's all for translation.
Thank you. Someone said in the chat is transcription where we just turn it into the RNA instead. And I think lectures, I think I found the lectures for that, but I think it's like 17 and 18 for
Oh, is it? Okay. Yeah, it's like taking DNA and making it into RNA. And then like for RNA to leave the nucleus, or like, yeah, to leave the cell, we need to make it into, or it has to leave as messenger RNA. Proofreading. This is like the end joining stuff that we have later. Oh, this this is very complicated. Oh yeah, this is transcription. Okay. RNA versus DNA. Oh yeah, these microRNAs, small RNA, or small interfering RNA, and micro and siRNA, they come in later for post-transcriptional gene silencing. Yeah, basically RNA uses ribose and then it's GACU. Wait, we're going to look at transcription. Oh, but we also need to know like what RNA does, so we can go over that. So yeah, so in the ribosomes, it's made of RNA, not proteins. So RNA does most of the work in ribosomes. And then the RNA world hypothesis is that RNA can self-catalyze chemical reactions. And these RNA are called riboenzymes. The hypothesis is that like RNA is the first form of genetic information on Earth. RNA can do a lot of things. Self-catalyze chemical reactions, transfer information, regulate gene expression. Transcription also has initiation, elongation, and termination. So to start transcription, RNA polymerase II, the transcription initiation complex, or is the pre-initiation complex, PIC, is set up. So a bunch of proteins come together. They're like general transcription factors. And then they like assemble into the PIC. So how transcription is controlled? So RNA transcription starts at the TATA box of DNA. A TATA binding protein binds to the box, changing the structure of DNA. And then transcription factors come in, followed by the RNA polymerase, and then they form the pre-initiation complex. And then RNA sequence is elongated by RNA polymerase. And then it's created in the 5' to 3' direction. And then once the polymerase reaches the termination site, the RNA is not lengthened anymore. Since RNA is very fragile compared to DNA, it's less stable. So we have RNA processing to do. So first thing that is done, or like one of the things that is done, is a 5' cap. And that's a methylated guanine cap. And then we have a 3' poly-A tail, which adds a bunch of U's and A's. Basically slows the degradation of the RNA. So like when nucleases come and eat the RNA later on, it slows that process, and mRNA can keep its message intact for longer. And then there's splicing, which is introns are taken out, they're cut out, and then exons are left in. Yeah, so modified guanine. Yeah, okay. Yeah, UTRs are just untranslated regions. They don't code for anything. There's UTRs on the 5' end and 3' end. Splicing just removes the whole thing. Like a loop is formed and then the loop is like taken out. Yeah, this is the 5' UTR, 3' UTR. Zip code basically allows the mRNA to like leave the cell later on. Then coding sequence makes like amino acids during translation. Yeah, and then the poly-A tail, a bunch of U's and A's. Now we got histones. Yeah, and then let's learn about the promoter. The promoter is on the 5' end of the gene, and then proteins bind. Or they're like attracted to the promoter. And then that makes RNA transcription start, because when proteins or transcription factors are attracted, they kind of come together into the pre-initiation complex, and then transcription starts. And then the terminator region stops transcription. Yeah, and then like other than promoters, there's like silencers, enhancers, and insulators. We didn't really talk about silencers and insulators, but for enhancers, they're usually, wait, can I get this to work? Okay. This is the promoter. This is backwards, but enhancers are like farther away from the promoter. They like they attract transcription factors, or like they guide them to the promoter. And then once there's a bunch of transcription factors of the promoter, transcription starts. Wait, that was the other slide. Okay. I don't know what's wrong with this. Yeah, okay. I think I talked about that. Okay. Oh yeah, and there's also a cat box. I don't know, there's a lot of boxes. But like cat and Tata box, we need to know those. The Tata box helps position RNA polymerase at the correct place on the DNA. It like guides polymerase like where to start transcription. Oh, I don't know. We talked about the cat box. Let's see. No, it's just like a binding site for transcription factors. And then they like form a complex, and then RNA polymerase does its job. Yeah, and then like promoters sometimes are like specific to certain tissues, or they can only be expressed in certain tissues. Yeah, this is like the enhancer. It's like far away from the promoter. But like the DNA strand kind of bends. So then like transcription factors are like attracted. And then for enhancers, they have an important job. So there's this gene called Sonic the Hedgehog, and it's regulated by the enhancer called ZRS. And then when ZRS is placed into a mouse, there's no like observable, not like nothing is observed. But like when it's when a snake ZRS is placed into a mouse, the mouse has limbs. So that means snakes used to have the enhancer ZRS, but they kind of lost it over time. And mice, they still have the enhancer, that's why they have limbs. Okay, what else should we talk about? Oh, meiosis, mitosis, and meiosis 1. That was like at the beginning of the year. Pedigree. Oh wait. It was like 12 or something. Oh, histone methylation and phosphorylation. Okay. Yeah, we can talk about that. That was like that was like 23 or something. Let's see. Um, what is that? Okay, wait. Oh yeah, we probably need this later. We gotta talk about the human genome and like percentages. Okay, I'll remember that. Um, maybe it's farther earlier. Okay. Yeah, like 22. But yeah, just talk about that. So 23. RNA-seq. No. Wait, let me actually look. I feel like it wasn't that long ago. Oh, regulating gene expression. That was November 12th. Oh, wait, no. It's November 10th. Watson, he did a lot of stuff. Chromatin. Oh, this is like the histone stuff. Okay. Well, yeah, we are talking about histones. So yeah, there's chromatin, and then chromatin wraps around histones. And then that determines if there's transcription or not, or like if the gene is expressed. And then the nucleosome is just 8 histones together. There's 2 pairs of each type. And then so we have like modifications. Histone modifying enzymes. Well, let's make this bigger. So we have histone deacetylases and histone acetyltransferases and histone methyltransferases and protein kinases and protein phosphatases. Yeah, okay. Anyway, there's histone demethylases. Yeah, we just need to know like the categories. So these 2 are for phosphorylation, these 2 are for methylation, these 2 are for acetylation. And then so histone acetylation is like the addition, like acetyl groups are added onto lost my train of thought. Okay. Oh, acetyl groups are added onto amino acids. So then, yeah, they're added onto lysine and arginine. And then acetylation is done by histone acetyltransferases. And that reduces how well DNA is attached to lysine or arginine and increases the transcription of DNA. Since lysine is positive, wait, in the resins, okay, sorry, how do I word this? I wrote it down somewhere, okay. So, so like the histone tails are made of amino acids, or they have amino acids on them. And then, so histone heads, they come together. Better if I draw this, okay. So, we have a histone head, another histone head, another one. There's like 8 of them, I'm not gonna draw 8. But then there's like tails. And these tails, they have amino acids on them. I'm just gonna put like AA. It's full of AA, full of amino acids. And then they attach to DNA. Or they wrap around DNA. So DNA, yeah, they wrap around DNA. Okay. So then if you add an acetyl group, or since like, okay, so DNA is negative. DNA is negative. Has a negative charge. Okay, DNA has a negative charge. And then the histone tails. Wait, DNA has a negative, wait. No, that's not important, okay. So the histone tails, when during acetylation, they go, like when you add a acetyl group, they go from a negative charge to a positive charge, or no, not positive, to a neutral charge. Because a methyl group is, or the acetyl group is like positive. Acetyl group is positive. So then when the histone tails have a neutral charge, they cannot wrap around DNA as well. So that means transcription goes up. Because when DNA is tightly bound or tightly wrapped, gene expression or the gene is not expressed very well, or gene expression is reduced. Yeah, there's basically this diagram. No, never mind, don't look at this diagram, it's confusing. Okay. Yeah, just remember this. Okay. And then when you remove a acetyl group, which is deacetylation, remove acetyl group. Yeah, spell that wrong. Remove acetyl group. HAL has a positive charge. HAL goes back to being positive. Histone, HAL goes back to being positive. And then that, oh, and then here's where like DNA being negative is important. So, okay, DNA has a negative charge. And then, for if histone histone tail is positive, it wraps tightly around DNA. And oh, what did I write here? This is for deacetylation. Okay. Wait, so if it has a neutral charge, DNA is not tightly wrapped. And gene expression increases. This is for acetylation. This is for that, and this is for this. Okay. Histone tail is positive, wraps tightly around DNA, and gene expression is reduced. Or transcription is reduced. Okay, let me add another page. For methylation and phosphorylation, those two are not like not definite, I think. Like you can't say for sure that they might cause expression, gene expression, or reduce gene expression. They sometimes increase or decrease transcription. Well, let's go back. Okay. So methylation is done by histone methyltransferases, and the demethylation is done by demethylases. Oh yeah, basically all these like H8 or histone acetyltransferases, methyltransferases, they all end in ASE. They're enzymes. And these, yeah, they do like modifications to histones. Okay, so the addition of a methyl group changes protein or how the protein folds, how a protein folds, and it also changes the interactions between proteins. Between proteins and DNA. So it basically changes how the histone tail attaches to DNA. I mean, all the modifications you get. Okay. Yeah, and then that's it for like methylation. Like we can't really say if it will increase or decrease. Like some of them do, some of them don't. It just depends. That's all we need to know. Yeah, and then phosphorylation is like the same thing. Phosphorylation. Oh, and so adding a phosphate group increases or decreases how well proteins are bound to DNA. Proteins in this case would be like the histone tails. Well, the histone tails are full of amino acids, and then together amino acids they make proteins. Yeah, and then that's it. And then we can go over like how to read or how to read like a specific histone modification. Okay, wait, no, that's not supposed to be a subscript. It's just number. Okay, this is just histone number 3. this would be the amino acid lysine. We don't need to know like what amino acid it is. We just need to know it's an amino acid. Amino acid K. But in this case, it's lysine. And then 8 is like the number or the position or 8 amino acids away from the tail. So we have a tail. It goes from like 1 to all the way to like 8. Or like, I don't know, like 100 or something. Depends on how many amino acids. But the 8th amino acid, or like 8 position, or 8 amino acids away from the tip of the tail, tip of the histone tail. And the 8th amino acid happens to be lysine. And then this is methylation. This is methylation. The modification, the histone modification is methylation. And then the 3 represents how many methyl groups. So number is, wait, 3 methyl groups. With 3 methyl groups added. And then like for the other modifications, acetylation would be AC. And phosphorylation is P. I think it's just a lowercase p, capital P. I don't know, it's just P. Yeah, and then that's it. Yeah, that's it for like histone modifications. Okay. Let's just make another one. That's it. Okay, let's see. Oh, wait, not that. Wait, I was not gonna look for meiosis. Okay. Meiosis. X inactivation. Not that. Okay. Do you have any other question? Oh, CRISPR? Okay. That's like the recent slides. Let's see. I remember there was 2 lectures on it. Maybe not. Okay, I'll just go the latest. So CRISPR technology helps with the or can help or might help with a lot of things in the future. Like it can help us modify genes and get rid of like the cancerous ones. But then there will be less diversity maybe. So CRISPR does gene silencing and gene editing on a larger scale. So CRISPR stands for clustered regularly interspaced short palindromic repeats. It uses repeats to basically match the RNAs of invaders and then like target those mRNAs and get rid of them. So CRISPR is a natural defense system against invaders like viruses in bacteria and archaea. And CRISPR is working to get like the system in bacteria and use it in other cells like eukaryotes. So far that has worked. Like CRISPR used in eukaryotes have worked so far. Okay, so this is how it works, and I'm gonna talk about it. I wrote notes. Okay. Oh, and I missed a point up here. Yeah. Okay, so in CRISPR, there's like Cas proteins, the CRISPR associated proteins, and then there are cRNAs or small RNAs transcribed from the CRISPR genomic region. Just like this locus CRISPR genomic region. And then the cRNAs, they guide the Cas proteins into targeting invader RNA. Yeah. So they match. So yeah, so like the RNAs, they guide the Cas proteins and then they match invader RNAs and then they cut the RNAs. Just like how dicer proteins and the RISC complex works. So in the late 1990s, Japanese researchers, they found a region in E. coli that has repeating DNA sequences. So these repeating sequences, it'll be like, repeats will be like CAG, 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 it keeps going. They didn't know what these repeats do. And then later on, we found out that upstream of the CRISPR region, or later on, we found out that the repeats actually have a function. And we also found out that upstream of the CRISPR locus, there are Cas genes that are being made. And then if we look at the sequences of these repeats or these spacer repeats, we can see that they match pass invader sequences. It's like a memory bank. 24? Oh, wait, I just saw that. Human genome. Yeah, there was something about alpha missets. I don't think it's in this one. Okay, anyway, I was on 26. I read that late, sorry. Okay. So these repeats, they have areas on them that match each other, and then they form a stem. This is the stem. This black loop, lollipop shape. Two sections of the RNA will loop against each other and match each other. And then they form these like small RNA sequences. Or yeah. And then the RNA sequences between the stems match bacteriophages. So just like how the dicer protein takes double-stranded RNA and then cuts it into small RNAs to target invaders, or no, target mRNAs and cut them. This is the same thing. Yeah. And then once the RNA sequences are created, another enzyme will cut here, cut off a part of the stem. Enzymes will come in and cut off a part of the stem. And then each of the segments, after these segments, they will contain the genetic memory of previous invaders of the bacterial cell. So in the past, like, bacteria might have encountered viruses attacking. Yeah, and then CRISPR just creates a genetic memory of that. And then these segments, they will associate with these Cas genes and guide the Cas genes into cutting the mRNA and then stopping the bacteriophages from replicating. Because if their RNA is cut, then they have no way to replicate or like inject DNA or inject genetic material into other cells. Yeah, and then this whole CRISPR system is like the bacteria's immune system. Oh yeah, the cRNAs are also called guide RNAs. Yeah, okay. So CRISPR, there's two uses in the lab, gene knockout and gene knock-in. Knockout is where expression of a gene is permanently prevented. Like, the gene is basically non-functional. It doesn't work at all. Not like how, what's it called? I just talked about it too. Okay. Oh, how RISC like silences the gene. CRISPR makes the gene non-functional. It doesn't work at all. And then there's also gene knock-in, where a gene sequence is replaced with an alternative sequence. So basically, there's like this target sequence, and then we provide a sequence, and then hope that like the DNA repair mechanism takes the sequence we provided and input it into the sequence that needs to be fixed. Okay, so for knockout, we make a guide RNA, and then it matches the DNA sequence, and then the guide RNA pairs up with the Cas protein, and then they make a double-stranded break. So this. And then the double-stranded break is repaired by non-homologous end joining. And non-homologous end joining basically has a bunch of, or no, has like, it's a sloppy way of fixing the gene. So then there's a bunch of errors, and basically the gene is non-functional. Because there's too many errors for it to work correctly. So yeah, and then for gene knock-in, we do the same thing. We make a guide RNA, we pair it up with a Cas protein, we make a double-stranded break, and then this time we provide a new sequence of gene, or new gene sequence into the system. Like we insert a new sequence. Oh, I should make this bigger. So we insert a new sequence, and then we hope that like homolog I think it was homologous directed repair. It like takes the sequence as a template and then makes another sequence based on what we inserted. And then the whole strand is like repaired. That's the hope though. But sometimes like the repair mechanism doesn't work at all, or it just uses the original homologous strand that it had from like when the sequence was broken. It like just uses the other strand, when that's not what we want. We want it to use the strand we inserted. That's why knock-in is more difficult than knockout. Yeah, this is just basically a summary. Yeah, and then yeah, so CRISPR, there will be a lot of breakthroughs in the future. So why we use why we should use CRISPR in the lab and why we shouldn't? Well, CRISPR might cure some genetic diseases, like Huntington's disease. It's cheaper compared to like other options, and it works in a lot of organisms. Like in most eukaryotic cells that we used it in, it worked. And then for knock-ins are not really like successful. They don't happen like all the time. And then there's like off-target effects. So off-target effects are basically like, maybe this gene is modified, but after the modification, like the patient gets cancer or something. That's like really bad. There's like side effects. And then there's like improvements on gene knock-in, which is homology independent targeted integration, which uses non-homologous end joining to insert like sequences. I think that was it. And yeah, so like oh yeah, so for Knockman, it uses like homology directed repair, right? So this HIT basically uses or does modifications and then it uses non-homologous end joining instead for the DNA we inserted. Oh, okay. Bye bye. Yeah. And then for base editing, so you can do an individual base mutation. So you can turn a C into a T without cutting DNA at all. And then for medical advances for CRISPR, so treatment of blood cells for sickle cell disease and beta thalassemia. So the patient's blood can be treated outside the body, then re-injected once it's deemed safe. One treatment is exa-cel that works by triggering fetal hemoglobin. This treatment was approved by the FDA recently, actually. And then for cancer treatment, involves modifying the patient's own immune cells to make them better at recognizing cancer cells, a form of immunotherapy. And then HIV treatment is a research program to use CRISPR technology to specifically target the HIV virus, removing it from the patient. And then, like, the other side of CRISPR, so germline therapy, in 2018, this guy, he modified the CCR5 gene of 2 human baby embryos. The babies were born healthy, but too early. He was sentenced to 3 years in prison because he illegally modified genes. And then now he's studying muscular dystrophy. That's why CRISPR can be a little scary because like you can modify humans and affect how they grow. But CRISPR might be able to treat Huntington's disease. But it might be too expensive once it becomes more the more it becomes used in the future. Yeah, that's it for CRISPR. Okay, now let's talk about meiosis. I can find the slides. Okay. Or I can just go from the top. I'll just go from the top. So why we should study genetics? Genetics is an important part of all biological studies. There was a lot of Nobel Prizes. Genetics, like data grows over decades. And as it becomes central to our daily lives in like medical advances, COVID-19, worldwide issues. Genetics has a potential for great advances in like solving sickle cell anemia or Huntington's disease. Why we should study genetics? Well, geneticists have a potential for great danger because there's like a bunch of clinical trials and whatnot that people have to test in order to make sure that their treatment works. Okay. Okay, so slide number 2. Before genetics, people wondered about heredity. Hippocrates thought that men and women both produce semen, and life is formed by the mixture of semen. And Aristotle thought that men produce the seeds of life. The seeds grow on menstrual blood. And Montaigne, I don't know why I'm pronouncing his name like that. Okay, let me pronounce it right. Montaigne, okay. Montaigne thought that traits such as disease can be inherited separately. So by the 1800s, plant and livestock breeders were interested in heredity, especially this guy named who was a abbot named Cyril Knapp. He basically recruited Gregor Mendel into his church, and then Mendel studied inheritance and heredity. So the two plant breeders that paved the way for modern genetics was Knight and Goss. They were both studying edible pea, which has the advantages of a short generation time, and the pea species has a lot of varieties, and the pea can cross-fertilize and self-fertilize. Cross-fertilize, which is transfer pollen from one plant to the ovule of the second plant. Yeah, you take like the anther and then put it onto the anther of another plant so that the pollen falls all the way down to the ovules. And then self-fertilization is taking the pollen of one plant to fertilize its own ovules. So the pollen on the anthers basically are like taken off and put into the ovules. This is what Mendel observed when he crossed a green pea plant with a yellow pea plant. And then blending inheritance is just like mixing of colors, like red and yellow, white equals red or pink. But that was not what the pea plant showed. So Mendel was the father of genetics. In 1990, William Batson, he discovered like a manuscript of all Mendel's experiments, and he was very interested, and he became like a follower. Mendel brought biology methods that were standard in physics. He limited the number of variables. He only used pure breeding plants. And he recorded the quantities of his results and came up with a model that could be tested. Okay, sorry. I was just pulling up my notes. Okay. Okay. So yeah, so yellow color was the dominant in F1 generation, but then afterwards the green phenotype or the green color resurfaced. Mendel performed reciprocal crosses to determine if a character was linked to sex or if one parent contributed more to progeny than the other. So he basically flipped the genotypes. So he crossed using one or crossed made a cross with one specific phenotype, and then he switched them and crossed it again. Sorry about that. Yeah, so the Mendel's true breeding plants are just true breeding are like they produce the same genotypes over a couple generations. Oh shoot. Okay. My paper is right. Okay. Mendel was aware. Oh shoot, never mind. And then phenotype is there's phenotype visual characteristics. Found out that the for this cross he found out that the flower color trait is not sex linked. Sex linked means the gametes, like egg and sperm, Non-gametes are somatic tissues. Since the hybrids were always purple, both parents must have contributed equally, so the flower color trait is not sex-linked, like Mendel found. Or that's what Mendel found. Okay, so he redid the non-crosses, but then he counted the progeny, and he got this ratio. It was a 3-to-1 ratio of yellow to green. He developed a model based on the ratios. Okay, lecture 3. So, so he did like this. So each somatic cell has 2 alleles, and if they're both the same, they're homozygous. If they're different, they're heterozygous. So he crossed yellow with green, and then he got a heterozygote, and then he crossed 2 heterozygotes, and then he got a 1-to-2-to-1 phenotypic ratio, but a 3-to-1 genotypic ratio. He did like a test cross. Wait, no, test cross is here. So a test cross is used to identify the genotype, if it was heterozygous or homozygous dominant of a particular phenotype. A test cross involves a cross with a homozygous recessive individual for the trait of interest. So then, yeah. A dihybrid cross means that there's like 2 sets of alleles for both individuals. And then you get like a 9-to-3-to-3-to-1 phenotypic ratio, and then a genotypic ratio is we don't need to know that. Okay. For crossing heterozygotes, it's 9-to-3-to-1. Okay, for product and sum rules. So probability is the number of times an event is expected to happen over the number of opportunities or trials. So probability of flipping a coin, or probability of flipping tails. Flipping tails is equal to 1 divided by 2. There are 2 chances, or there are 2 outcomes, but only one outcome is flipping tails. So, if we want to find the probability of this cross, we just list out the possible gametes, and then we do the probability of how many times the gametes appear. So probability of capital R, capital Y, that's 1 time, and there are 4 possible gametes, so that is 25%. The lamb product rule is the probability of independent events occurring together. Is the product of the probabilities of individual events. So the probability of tail and head. Of flipping a tail and a head equals 1/2 times 1/2, which is 1/4, which is 25%. So then what is the probability of finding a zygote of RRYY genotype from 2 heterozygous parents? So we 2 heterozygous parents, we have RR, YR, or YY times so we can choose one trait to look at so it doesn't get confusing. We can choose R first. And then we want to get RRYY. Whoops, okay. We want to get RRYY. Wait, what's this is capital? That is a horrendous Y. I mean, horrendous R. Okay, anyway. So probability of getting a double homozygous dominant or a homozygous dominant is 1/4. Now next we can look at Y alleles. We're also gonna get 1/4. So the probability of the genotype capital R, capital Y is 1/4 times 1/4. Which is 1/16. Okay, and then yeah, that's that. And then what is the probability for this genotype? If we have these parents. Oh yeah, I can't edit this, so okay. So let me split screen. Okay, I can see better. Oh, we have AA, B, B, on AABE. We can start looking at the A genotype. Oh, this is lowercase. How do we erase? Big eraser. Okay. I discovered I can erase this whole time. Okay. Was A, capital lowercase a, and B, and then B. So then we have A, no, we have A, and then we want to get A, A, B, B. So we have A, capital A, capital A, capital A, capital A, lowercase a, or capital A, lowercase a, capital A. Got some messy writing. Okay, anyway, this is 1/2. And then for BB, it's also 1/2. So it is 1/4. One fourth. There's a probability. And now onto sum rule. Product rule, you multiply, and it is and. Product rule, multiply, and keyword is and. And then the sum rule, you add, and keyword is or. So we have a parents, heterozygous parents. There's 2 of them. I'm too lazy to write it. Okay, so there's 2 of them. Okay. No, it's just because I'm writing with my mouse and it's kind of hard. Okay. And we want to see if the probability of this phenotype or this phenotype. That is a wacky R. Okay. Anyway, and then we can do cross with the genotypes of the parents. R. I apologize for my messy writing. And then we can do. Oh, this is lowercase. Cross for the Y's. It's the same. Okay, anyway, and then so we want to get homozygous dominant, and there's only one in this cross. So 1 out of 4 homozygous dominant. Then we got heterozygous dominant, and that is 1/2. And then we want homozygous dominant for Y's, that is 1/4. And then we want homozygous dominant for our homozygous recessive, and that is 1/4. So the probability of this probability of that is 1/4 plus 1/4. Wait, am I doing this right? Oh, wait, first we gotta multiply them. So cross that out. Okay, so first we gotta multiply them like the previous one. So we first gotta do product rule for like the R and Y. Probability of R and Y. So it's 1/4 times 1/4 equals 1/16. And then the other one here is 1/4 times 1/2. Then we do 1/8 plus 1/16, and that equals 3/16. Or the probability of homozygous dominant R, homozygous dominant Y, or heterozygous dominant R, or recessive or homozygous recessive Y. This is the probability. 3/16. Sorry, it's so messy. Yeah, and then the other ones are basically the same thing. Even this one. You just split it up. Yeah, let's do this one because it's kind of complicated. I'm gonna type. Okay, we have this. So we do three separate tables. Wait, these two are lowercase. It just looks kind of weird. Lowercase, capital, lowercase. Okay, so we want the probability of smooth red fruit with purple stem. Okay. Smooth red. P P A A R R something like this. Yeah. Okay, so or this A can also be heterozygous. This R can also be heterozygous. So the P's, we don't have any homozygous dominant, so we have these two out of 4, which is 1/2. And we have A, well, A. And that is 3 out of 4. And then the last one, R. This is supposed to be capital. It's 100% all purple. 100% purple, so it's 1. And we add all those together. We're not adding, we're doing the product rule for this one. So we're gonna get 3 out of 8. Since it's not saying or anything, we're not given like 2 of these. So it's just 3/8. Okay, and then number 5. Okay, number 5 is pedigree analysis. D. This one kind of difficult. Okay, so recessive traits appear in the progeny of unaffected parents. They show up in these kind of matings, which is like matings between relatives, and then all progenies from two affected individuals are affected. So yeah, and then the 3 steps is establish genotypes in the situation that the child is affected, then determine the probabilities of each genotype, and then apply the product rule to determine the overall probability of the child being affected. So that's how he did it, but okay, let's split view. So we have an individual, another individual, another one, another individual with siblings. And then the last individual is a child, which is a diamond shape. And then if the child was affected by the disease, they would have a recessive allele, dominant recessive genotype. These two are the affected, so they also have recessive. So in order to get recessive, these two parents need to be homozygous. And these two parents also need to be homozygous if the child was got the disease. And then this individual, since they're outside the family, they're gonna have the homozygous dominant phenotype. This individual is inside the family, and since the parent here is heterozygous, this lady needs to be heterozygous so that we can have this phenotype. And then same thing for the top two parents. This individual is also from outside the family. And then this individual, we don't really need to know because they're not passing down anything. Only the mother is passing down the disease allele. And after, we need to find like the probability of all these alleles or these genotypes. So this one on the bottom is just a homozygous times homozygous cross, or heterozygous, not homozygous. Homozygous. And we're gonna get 1 out of 4. And then if we do a cross again for these two individuals, we're gonna get this the same pedigree, but we have to cross out the recessive one because we know that this individual is not affected. So if they're not affected, then the recessive does not apply, so we gotta cross it out, and it's gonna be 2 out of 3. We don't cross out this one because we don't know for sure if it's if this individual is heterozygous or homozygous dominant. And then if we do A, let's cross. We're gonna get 1/2. This individual has probability of 1 because we know for sure that they have the disease. This individual also has 1 because we know for sure that they're gonna not have or they're not gonna be affected, so they're gonna be heterozygous. Because there's no way they can be homozygous dominant since this parent is affected. And then this individual we don't care about because they don't really pass down any disease alleles. The same with this individual. But then if we do another cross, another cross with a dominant, homozygous dominant and heterozygous dominant cross for these two, we're gonna get the same 1/2. Now we don't really care about these two individuals because they don't really contribute to the disease. So that's it for the probabilities. I'm just gonna do one probability problem because I already talked about some in the other video. So now let's go into lecture 6. Oh, this is also pedigrees. But it's about like disorders and autosomal recessive means that affects individuals with recessive alleles. And then same with this disease. I don't have recessive. Okay. Number 7. Gene interactions. Well, incomplete dominance is like basically like a mix of the two alleles. Like pink and red, red and white flowers, they make pink. There's like a middleman, there's a middle allele. So long and round carrots, they're gonna make oval carrot. But it still follows Mendel in some way. There's still 1 to 2 to 1 phenotypic ratio. And then codominance is basically like spots, like polka dots. Like if you have white and black, you're gonna get like a spotted, you're gonna get spots. So like if you have a black chicken and a white chicken, you might get a spotted chicken. And then sickle cell anemia. Wait a sec, give me a second. Sickle cell anemia. Okay. So yeah, hemoglobin is responsible for transporting oxygen, and a variant of hemoglobin, HbS, results in the aggregation of hemoglobin, causing the cell to change shape, so it changes into the sickle shape. So heterozygotes have both phenotypes. They are round and sickle cell. So this is heterozygous. And then HbS is sickle cell, and HbA is round and not affected by sickle cell anemia. That codominant is like this, incomplete is like a mix of colors. Okay, then we have duplicate genes. So red times a white flower equals red. But then in the next generation, you have a couple white flowers, and then like majority is red. And then the phenotypic ratio is 15:1. That doesn't really follow Mendel's rules, but yeah. The two genes A and B, whose functions are generally the same, can modify the observed phenotypic ratio compared to a single gene case. Oh, and no, they're not a violation of Mendel's model. Never mind. So duplication is not a violation. Yeah, so why duplication happens is that there are two sets of alleles for the same gene. So incomplete codominance and duplicate genes, they change the phenotypic, not genotypic ratios. Oh, so it still follows Mendel like that. And then, yeah. And then multiple genes, they often affect a single trait, like skin color. Because in skin color, at least 100 genes are involved and they all act independently. This seems like blending inheritance, but it's not. Blending is like two colors mixing together and then you get like a overall color. But no, it's not like that. Then Mendel's model does not apply because there are like a lot of genes involved. And then we have epistasis. Which is basically there is one color that's on top of the other colors. The other colors. So like the hair follicles of these mice, there are 3 genes involved. And then the agouti color is mm, okay, I don't okay. Yeah. So like when you do a cross, gene C, which is agouti, basically affects the expression of cinnamon and albino colors. So the ratio is 9:3:4. And then there's also a case of multiple alleles. Oh yeah, epistasis is when the expression of a trait is dependent on the expression of other traits. One gene or several genes can stand over other genes. It's also an example of multiple genes affecting a single trait. So, and then multiple alleles is like blood type or blood groups. 6 possible genotypes can occur because there are multiple alleles. Types A and B refer to different sugars on the red blood cells. So blood type A has 2 phenotypes, blood type B has or 2 genotypes, and then A, B, and O only have 1. Yeah, you could also do pedigree for this. Multiple alleles. There are more than 2 alleles for a single gene in the population. Any individual can have 2 alleles. But his model still holds true in this case. Okay, meiosis and mitosis. Okay. Give me a second. Okay, mitosis and meiosis. I'm just looking like what videos I posted for the lecture. Okay. Oh, 10 to 13. Okay, I posted that, so I don't need to talk about that again. But okay, meiosis and mitosis. So Walter Fleming investigates cell division and describes chromatin as thread-like structures in the cell nucleus. Yeah. So chromatin is made up of chromosomes. And Fleming described how these are involved in a type of cell division called mitosis. Mitosis is a type of cell division where the end results are two genetically identical diploid cell daughter cells. Okay, the number of chromosomes are variable. Diploid number is 2n, haploid is 1n. Humans have 46 chromosomes, chimpanzees have 48, fruit flies have 8. In diploid organisms, though chromosomes are in pairs, humans have 23 pairs of different chromosomes. The pairs are homologous chromosomes. Yeah, okay. So chromosomes are duplicated during the cell cycle. during the S phase, which is also called DNA replication. And there are sister chromatids. The pairs of chromosomes are homologous chromosomes, and individual ones are sister chromatids. So mitosis, two identical diploid daughter cells. We have interphase, which we just have a cell, and then the nuclear envelope dissipates, and then prophase. The chromosomes basically are like compact into chromatids, and then prometaphase. The nuclear envelope. The nuclear envelope breaks apart, and then like microtubules attach to the chromosomes to pull them apart or to place them at the center in metaphase. And then in anaphase, the microtubules pull apart the chromosomes into, like, they separate the chromosomes into halves. And then in telophase, basically the cell is cleaved and we get two separate cells, and then cytokinesis, the two cells become, or the cleaved cell becomes independent, or creates two independent daughter cells. And then a special division for gametes, Bendon observed in a system he was studying that egg and sperm cells contain 2 chromosomes while adult cells contain 4 chromosomes. Wiseman predicted that there must be a special nuclear division which reduces the number of chromosomes in half in gametes. And this is meiosis. Meiosis, copy of each type of chromosome is sent into gametes. Gametes fuse and restore original diploid and number of chromosomes. A single cell, okay. Oh, that way when gametes fuse, you restore the original diploid number of chromosomes. Okay. So meiosis is kind of like mitosis, but you have prophase 1, centrosomes, and it's just a cell, and then later prophase 1, a chiasmata or tetrad complex is formed, and then chromatids share genetic information. And then at metaphase 1, the chromatid complex is brought to the middle to form this plate, and then anaphase 1, the chromatids are pulled apart, and then telophase, the cell is cleaved, and then the cleaved cell separates into 2. Yeah, parental chromosomes are bound together as a tetrad complex made of 4 chromatids. And they recombine, go through recombination to share genetic information. And then meiosis 2 is separation of chromatids. Oh wait, in meiosis 1, there's no cytokinesis, the 2 cells just go into meiosis 2. And then, yeah, the chromatids are compact again. They're brought to the middle, they separate, and then you have 4 cells. and then gamete production is meiosis 2, and then it becomes sperm cells if you start with a spermatocyte, so diploid to haploid, and then for oocyte, diploid to haploid, but then you have polar bodies, and then the polar bodies are basically thrown away or like reabsorbed or whatever, and then it becomes one egg cell. So outcome of meiosis, reduction, division, one cell with two genomes becomes four cell with one genome. The four cells gametes produce contain half the number of chromosomes found in the premeiotic cell. Genetic recombination, the chromosomes now contain a scramble of the genes that existed in the parental chromosomes due to crossing over, chiasmata. If crossing over occurred, they are no longer identical copies of the same DNA molecule. Each cell contains one copy of the genome, making it haploid instead of diploid. The chromosome theory of inheritance. These two realized that chromosomes behave like what Mendel talked about. They come in pairs when the cells divide, and each member of the pair goes to different cells. And that chromosomes might be like Mendel's alleles. Okay, number 9. So fruit flies. Morgan studied fruit flies, and fruit flies have a short life cycle. They produce many progeny, and they're easy to grow in laboratories. Wild type is a allele type found in nature and is assumed to be dominant. Fruit fly genes are named after the first mutant allele discovered, not the wild type allele. So W plus is red eyes, W plus W is red eyes, WW is white eyes. Sex chromosomes. Males are unpaired or mismatched pairs of chromosomes, X0 or XY. Females are matched pair of XX. This is true for mammals, but not other organisms. The fly room. Morgan wanted to demonstrate the chromosome theory of inheritance. He needed a phenotypic trait that could be linked to a chromosome and that could be followed. What did he use? Let's see. He used the X chromosome. So he looked at a mutation for white eyes on the X chromosome. And he did some crosses. And more crosses. And got these results. And then he did another cross, test cross. Did a test cross and got these results. Then he did a reciprocal cross. And then the summary is the mutant phenotype white eye was inherited in the same manner as the X chromosome. This provided partial proof of the chromosome theory of inheritance. Yeah, okay. And then we have Calvin Bridges. He found that 1 in 2,000 times, he got red-eyed female or red-eyed male and white-eyed female. How is that possible? Because of recombination. Or, yeah. Or because, like non-disjunction, because somehow the flies weren't able to get the correct chromosomes. If a fly has 2 X chromosomes, it's female. If it has 1 X chromosome, it's male. But if it has 3 or 0 X chromosomes, the fly dies. And fruit fly sex is determined by the number of X chromosomes, not Y chromosomes. The presence of Y chromosomes means that the individual or organism is male. Okay, in humans, if you have XXY is a fertile female for flies. For humans, if you have XXY, you have a Klinefelter syndrome, and this happens in males. And then for flies, if you have X0 chromosome, it's a sterile male, and for humans, it's a Turner syndrome. Okay, the segregation of sex-linked genes in males requires pairing of chromosomes X and Y. Genes in the differential region of X are hemizygous in males. That means there's only one copy, and it's either X-linked or Y-linked. Genes on sex chromosomes are not necessarily involved in sex functions. X-linked genes are like color blindness. Color blindness is mostly in males because males only pass down a Y, or males pass down a Y chromosome. Or wait, let me actually, I forgot the actual reason. It was like something like that though. Okay. More common because color blindness is caused by genes on the X chromosome, and males only have 1 X chromosome. So if they have a defective X chromosome, males become colorblind, while females need 2 X's to become colorblind. Okay, now let's go to chapter 14, because I already did 10 to 13. So 14. Bacterial genetics and DNA structure. Let's look for that. So bacteria has a short doubling time, and under ideal conditions, they can reproduce in 20 minutes. They have a small genome size, So you can observe a lot of phenotypes. Wait, am I on the right? I don't know. Okay. I feel like I'm not on the right one. Oh wait, let's speak about biochemical pathways first. Okay, so the synthesis of amino acids, vitamins, or etc. occurs in steps. Mutations in any genes encoding the enzymes in a pathway leads to inability to grow without the appropriate supplement. The bacteria cannot complete a biochemical pathway. It's called auxotrophic bacteria. And then phototrophic bacteria can complete the whole pathway. So if the enzyme doesn't work, then we cannot make not make phenylpyruvate, and if there's no supplement, then the pathway just stops, and we cannot make phenylalanine, which is the final product. Without phenylalanine, bacteria cannot survive. However, auxotrophic bacteria can be rescued by providing them with an artificially appropriate biochemical. So you can supplement them. You can determine which gene is mutated in a bacterial colony by growing the bacterial plates with various supplements. Okay, genes are indicated using 3 letters not capitalized, followed by a plus for wild type prototroph. That means they can make a supplement or make whatever they need in the pathway, and then minus for mutant auxotroph. So they cannot synthesize what they need, and they need to be rescued. But plus they can make what they need. They don't need to be supplemented. We can also identify bacteria based on antibiotic resistance. Bacteria will not grow in colonies on the medium containing certain antibiotics. Streptomycin is an antibiotic that interferes with the correct initiation of protein synthesis. So that means bacteria will not grow on the medium with STRS. With STRR, bacteria will grow on the medium with the antibiotic. Same thing with ampicillin and antibiotic. If it's R, then you can grow. Bacteria can grow. If it's S, it can't grow. So how do we observe bacteria? We can plate them. We can see what colonies and single bacteria are doing through plating. So we take a minimal medium of broth with everything bacteria needs in order to grow. And put bacterial cells in the broth, and then put on a plate, and then leave it out to for the bacteria to grow and incubate it, and then you can see colonies. And then a second lab technique is replica plating. You can quickly transfer colonies from one plate to another. The original plate is the master plate. It is pressed on a piece of velvet, and then you can transfer 50% of cells from each colony. The second plate, the replica 1, is pressed on the velvet to leave an imprint. And then you can make a bunch of replicas. Velvet has spikes, so bacteria get stuck on the spikes or impaled on it. And then cells are incubated, and then you get new colonies of bacteria. You don't need to transfer a lot of cells with replica plating. So Lederberg and Tatum. Do bacteria have sex? Which is a mix of two genomes to form a new individual. They do. So they mixed. So they mixed two strains of bacteria. They plated it. Colonies showed up. But when they, yeah. But when they plated the individual strains, there was nothing. Then this shows that recombination happened. But other people believe that it wasn't really bacterial sex, it was just bacteria being really good at picking up stuff. So then the YouTube experiment is where they use the same two strains. They put a filter between the two strains and see if recombination would happen. But there was nothing, only debris went through the two strains. So that means bacterial recombination requires physical contact between the bacteria. Genes are transferred in one direction. Oh, that's the next one. Next one is genes are transferred in one direction. So they use similar bacterial strains. They treated strain A with a little dose of streptomycin. The strain A bacteria had sex and then died. And then he poisoned strain B with streptomycin, but there was no colonies. But then when they plated the strain A and strain B, colonies grew. This shows that strain A can donate genetic information to strain B, but strain B cannot donate to strain A. So fertility factor, F factor contains pili genes, transfer genes, replication genes, but no genes essential for cell survival or growth. Pili, bacteria can interact and fuse using their pili. So there are two types of bacteria, F minus, they do not carry the F factor in the cytoplasm. They do not contain pili on the surface. They cannot transfer DNA by conjugation, which is like forming a bridge, and then transfer genetic information. They can receive DNA transfer from F plus or HFR bacteria. F plus can carry the F factor in the cytoplasm, contain pili on the surface, and transfer F factor to F minus bacteria. So donor cells in Hayes' experiment A strain, cells that can donate DNA to other cells, F plus and HFR. Which is HFR is high frequency bacteria. And the recipient cells in his experiment was B strain, cells that can receive DNA from other cells, and F minus cells. The pili bring F plus and F minus cells together so that there can be an exchange of genetic information. And that is done through conjugation. After conjugation, the F plus becomes F minus, and then F minus becomes F plus. And F plus cells can often go back to F minus if they want to. DNA structure, okay. So was DNA or protein genetic information? DNA was first described by Meister, and most geneticists thought it was protein instead of DNA. So Griffith's transforming factor, then we have S strain and R strain. So we have S strain, you feed it to the mice, it causes it to die, but the non-pathogenic doesn't harm the mouse. But then when you heat up the S strain and mix it with R strain and inject it to the mice, it dies. So that means something must have happened to transform the R strain into S strain. Because heat denatures proteins and kills bacteria. So it was unlikely that proteins will still carry information after being boiled. But DNA, on the other hand, is more likely because DNA is like salt, because it still contains its original form after being heated. Oswald demonstrated that transformation factor was likely DNA. Okay, the Hershey-Chase experiment, they used bacteriophages, viruses that infect bacteria, and they labeled it with phosphate, radioactive tracers, phosphate for nucleic acids, and sulfur for proteins. Viruses inject genetic information in the cell's nucleus, and then the cell will make viruses, and then the cell will explode. Then viruses keep invading other cells. So then in this experiment, they grew bacteriophages in the presence of phosphorus. The heads of the bacteriophages would be radioactive, and in the presence of sulfur, the protein is radioactive. They washed the system and injected genetic information in the and the injected genetic information in the bacteria disappeared for sulfur. But when they washed the system for the phosphorus, the cells were still radioactive. That means DNA was injected in bacteria, not proteins. So in 1944, Schrödinger publishes a book on the nature of life and the concept of a genetic code. And then biologists finally accept that DNA is a source of genetic information. They come to accept. A DNA nucleotide has a phosphate, 5-carbon sugar, deoxyribose, and ribose sugars, nucleotides. Oh wait, ribose is for RNA. Then purines, pure has 2 gold rings. We have adenine and guanine, and pyrimidines. Cut A pi, cytosine, thymine, and uracil. Chargaff, he was a supporter of Avery's evidence that DNA was genetic information. So he analyzed the percentage of each of the bases and found that they were consistent within species and varies from species to species. So percent of A equals percent of T, percent of C equals percent of G. X-ray crystallography. Like a technique to find the 3D shape of a molecular structure. So you have a crystal lattice structure and you shoot an X-ray made of individual photons, and the photons will hit atoms in the crystal and deflect. The photon will hit a film and produce a dot, and then we analyze angles to find the structure. After DNA was genetic material, everyone wanted to find the structure of DNA. Several labs used crystallography to find DNA structure by turning DNA into a crystal. Rosalind Franklin and Maurice Wilkins performed this technique on DNA. Franklin created Form A of DNA, Form A and Form B crystals of DNA. Form A is outside of water, Form B is inside water. She worked on Form A a lot, but she didn't know that DNA was always in water. But eventually she worked with the right form of DNA. Francis Crick and James Watson, they collaborated with Wilkins and Franklin to produce a double helix model of DNA. And then in 1953, 3 papers were published about the structure of DNA. The model of DNA. The carbon atoms in organic chemistry are numbered. Left-handed helix. While most DNA helices are left-handed helices, a phosphate group is attached to the 5' carbon of the nucleotide, and new nucleotides attach phosphate to a free 3' carbon. The 2 DNA strands are anti-parallel. And then DNA has a phosphodiester bond right here. Is made of carbon, sugar, and phosphates. Sugar and phosphates creates a sugar-phosphate backbone. And then adenine and thymine, they have 2 hydrogen bonds, and guanine and cytosine has 3 hydrogen bonds. Hydrogen bonds are weak bonds, they break easily. Sugar and phosphates have covalent bonds, which are strong bonds. The phosphate diester bond gave Watson and Crick an idea of what DNA structure is, what the structure of DNA is. So how is DNA replicated? Watson and Crick proposed a mechanism for DNA replication. They suggested that a new molecule of DNA contains 1 parental and 1 newly synthesized. And that is semi-conservative model. So Mendelssohn and Stahl, they demonstrated Watson and Crick's replication mechanism. And Kornberg, he discovered DNA polymerase enzyme. Mendelssohn and Stahl, they use a nitrogen 15 solution to grow cells in, and then the resulting DNA would have 2 heavy strands. They spun the strands in cesium chloride and separated to tell how heavy the strands were. And then the data was consistent with a semi-conservative model. When isoleucine installed, they took isotopes of nitrogen, they labeled DNA with isotopes and made DNA heavier. Lighter DNA and heavier DNA would separate, and only the semi-conservative model data was consistent. Watson and Crick thought that there was an enzyme that makes DNA, or there was an enzyme that makes DNA. And then Kornberg found that the enzyme DNA polymerase takes a single strand of DNA as a template strand and makes a second strand of DNA. Well, DNA synthesis in bacteria. It was described first, bacteria have a single chromosome, circular chromosome. In the bacterial genome, origin of replication is where replication starts, and the area when DNA replication can occur is at the replication fork, and there are two replication forks in bacteria that work at the same time. Helicase unwinds DNA helix, breaks hydrogen bonds, makes DNA available for replication, and then tries to reach the termination of replication in the bacterial fork. The two forks have to meet at the termination of replication at the same time. If they don't, then there are signals that are sent in order for the two polymerases to meet at the same time. Yeah, that was bacteria. So there was a problem of large and linear DNA replication. And Tsuneko and Reiji Okazaki, they thought of Okazaki fragments. So one strand of DNA is made in fragments. It is not synthesized continuously. The leading strand is synthesized continuously, while the lagging strand has gaps. Helicase breaks hydrogen bonds between strands and opens up the DNA single strand binding proteins that keep the parental strands apart. Primase synthesizes primers to guide DNA polymerase on where to replicate, and then DNA ligase fills in the gaps between Okazaki fragments. So differences between bacterial and eukaryotic DNA replication. Eukaryotes have multiple replication forks. Okazaki fragments are shorter in eukaryotes than prokaryotes. Prokaryote replication ends at the termination site. Eukaryote replication ends at the chromosome where a telomere is located. A telomere is just like a set of 3 nucleotides that is placed at the end to basically make one strand longer. So when nucleosome comes, it just bites off the extra on the DNA strand. So there are more mutations on the lagging strand than the leading strand. So the lagging strand is believed to be the origin of some DNA mutations. DNA repair, real life mutants. They're kind of cute. Okay. So Muller was frustrated by the low frequency of fly mutants. So he thought that, so he figured out that radiation can produce more mutants, and bacteriophages would be useful to study in studying genetics. Fruit fly mutants have to be carefully researched, or at least to sterility or death. For humans, radiation exposure leads to death or cancer, but not mutants. Genome stability, there are 3 billion base pairs in the human genome. 2 copies of a genome per cell, around 40 trillion cells in the human body. There's a lot, so that means we need DNA repair. And we need a lot of DNA repair mechanisms. So there are many causes to mutations. Sources of mutations include radiation, DNA replication, which is the main error, chemicals, human-made or natural chemicals, UV light, which is the second error, byproducts of reactions, mobile genetic elements, jumping genes that move around, that move DNA around and cause damage. The side note, DNA damage from alcohol. The liver breaks down alcohol and acetaldehyde forms and bonds to DNA. Acetaldehyde is linked to gastric cancers. So how does DNA repair work? So there is proofreading done by polymerases. They check what they put down. There is repair doesn't need a template. There's a direct reversal. So a mistake is directly removed, and then a repair where template is needed. And then there's a repair of double-stranded breaks. Repair of direct reversal is when UV light for example, UV light causing cytosine or thymine to bind to each other, causing a dimer, a limiting dimer. And then photolase enzymes repair those dimers. But we don't have these kind of enzymes in humans, but plants. Direct reversal fixes specific damage. Repair of one-stranded template is mismatch repair. Repairs DNA replication errors missed by proofreading. And then the repair of double-stranded breaks, it's uncommon and high-energy breaks, such as radiation. Well, it's repair of uncommon high-energy breaks, such as radiation. There are 2 types of repair: non-homologous end joining, NHEJ. They join DNA without using the homologous chromosome as a template. And it's the most common, but it always makes a mistake because it's sloppy. It just puts stuff together. And then homologous recombination joins broken DNA using homologous chromosome as a template. So if there's a breaking chromosome 1, it uses another chromosome 1 to fix. And this technique is used in gametes, but it's a lot of work and a lot of enzymes and steps are involved. It's reserved for important DNA like gametes, not for somatic cells in humans. So there are many errors in DNA replication. DNA replication has an error rate of 1 in 10,000 bases. And the proofreading error brings that to 1 in 10 million bases, and mismatch repair brings that to 1 in 10 billion bases. The average human body still has 30 to 70 trillion cells, so there's still a lot of error. DNA mismatch repair covers mismatches of A, T, C, and G, and it maintains gene stability by repairing after DNA replication. The loss of mismatch repair leads to substitution and frameshift mutations. The loss of mismatch repair in proteins leads to cancers, heredity, like HNPCC. The 3 steps in DNA mismatch repair is recognition of the damaged site, removal of lesion by nucleases, destroys a whole section, resynthesis of new DNA strand, rebuild. So E. coli versus eukaryotic and mismatch repair. Okay. So the mismatch repair detects with a dimer, which is a diamond shape. And then two proteins surround DNA like a donut, and they are looking for bumps or dimers. The distance between C and C is longer than the regular pairing of G and C. Once they find bumps, a bunch of enzymes come and look for DATC sequences away from the error. They find the sequences and two proteins will come together and form a loop and destroy everything inside the loop. So they form a loop right here. The sequence will be rebuilt and the nucleus will be recruited and only destroy a newly synthesized strand. And the original strand is binded by single stranded binding proteins. And DNA polymerase uses original strand to remake bottom strand. In bacteria, the top strand has a methyl group, or CH3 right there, which is added after replication. Nuclease looks for a methyl group and destroy the one that does not. And then the strand discrimination problem is that in eukaryotes, there's no methyl groups on top of the original strand. So how does the body know which one to destroy or not? Oh, well, we don't know, but we could, or a suggestion is that there might be tiny errors that are put purposely in the new strand, and then the errors create gaps for nuclease to detect. I think I talked about this in the earlier. Oh, I talked about this also. Okay. Um, what is this? Oh, this is like the chi-square stuff. Oh, this is review. Whoopsie. But this is like the corn stuff. Okay, 20. I kind of did talk about histones also. Okay, 21. Okay, so DNA sequence can also be modified, repressing the expression of specific genomic regions. So we have CPG islands. Wait, I need to talk about T6. I didn't talk about that, so let's go back. T6 should be in here. But I already talked about methylation and whatnot. Oh, epigenetics. Epigenetics is something on top of genetics, and just saying that, like, just like explores which parts of the gene or which parts of DNA are expressed and which parts are not. Uses the same DNA sequence and you get different types by expressing DNA in different ways. Like X inactivation in calico cats, you have a different copattern, or agouti mice gene with 3 different mice colors, brown, black, or white. Or identical twins having different autism diagnoses. Epigenetics is reversible, and it's a study of reversible heritable changes. Okay. And then we can talk about T6. So long RNAs, non-coding RNAs, LNC RNAs, and X inactivation. So X inactive specific transcript X's inactivates one of the X chromosomes in female mammals. X's can be regulated by LNC RNA or T6. So X's sends a signal and then X chromosome is inactivated. During development, there is a signal that is passed on that inactivates one of the X chromosomes. The signal affects the X's gene. Once activated, X's sends a second signal to the other chromosome not to activate. The deactivation of the X chromosome is done by LNC RNAs. LNC RNAs are long RNAs, non-coding RNAs that do not code for proteins, that will wrap itself around the X chromosome so tightly and prevent the DNA of the X chromosome from doing anything, and then the DNA will shrink into a little ball, like polar bodies of oocytes. The system discovered the long RNAs and X inactivation was discovered by Dr. Caroline Brown and their lab. T6 and X6 are 2 signals on the same piece of DNA. T6 says do not deactivate X chromosome, and X6 says inactivate X chromosome. Okay, let's go to the next one now. So CPG islands. So CPG can be methylated to generate 5-methylcytosine. Reaction is catalyzed by the DNA methyltransferase enzymes. Wait. Okay, so DNA sequence can be modified, repressing the expression of specific genomic regions. Most mammalian genes have CG-rich areas of DNA in their promoter regions. These are called CPG islands. Methylation of the C residues within the CPG islands leads to gene silencing. CPG Island, short stretches of DNA rich in the bases cytosine and guanine. They're located near the start site, the promoter of genes, and help control a gene's activity. In mammals, demethyltransferases are involved in different types of methylation. De novo methylation produces a brand new methylation pattern and puts on both strands. Demethylation removes methylation patterns, and the other one, maintenance, let me see what it does. I have it on the last page. Oh, after DNA replication, we are going to replicate the same pattern methylation onto the new strand as we had on the old strand. That's for maintenance. So, expression of DNA sequences is influenced by their accessibility to transcription proteins, and this is studied through epigenetics. And DNA is packaged in histones, and then acetyl groups, methyl groups, and phosphate groups can be placed on the tails of histones to influence the interaction between histones and DNA. DNA can be directly methylated too. So the types of small RNAs, microRNA, siRNA, and piRNA. PTGS mechanisms, post-transcriptional gene silencing that happens after transcription, is specific to certain types of RNAs. Some mechanisms use more than one RNA. It uses double-stranded RNA, small RNAs combined with specialized proteins to accomplish their silencing role. The presence of double-stranded RNA activates RNAi or RNA interference or PTGS. PTGS mechanisms include cleavage of mRNA, breaking RNA in half and destroying it, shortening the poly A tail, making mRNA more difficult to be translated by ribosomes. So there's a lot of baggage attached. Like a lot of proteins attracted to the mRNA are attached to it. Okay, so dicer finds the double-stranded RNA detected and dice it up into small RNAs to carry around. And then the RISC complex with the central protein of Argonaute 2 or AGO2. AGO2, take the segment of small RNA and look for the target mRNA of past invaders so that it can destroy the target mRNA. For cleaving, there are 3 different types of dicing and making double-stranded breaks. We have cisnet RNA, transnet RNA, and extended hairpin RNA. So cis mRNAs form a double-stranded RNA and trigger PTGS, transcribing same location PTGS transcription. Okay, I don't know what I wrote, so I'm just gonna not say that. I don't think we need to know that anyway, so cleavage of mRNAs is only one way miRNAs, siRNAs, and piRNAs can regulate gene expression. siRNAs are favored in plants, but less in mammals who use miRNA. siRNAs are required to match the target perfectly, while miRNAs and piRNAs tolerate some mismatches. piRNAs specialize in repressing a type of genomic parasite called transposable elements. So bacteria use small RNAs too, like CRISPR or Cas9 system is a natural defense system against invaders. It's made up of cluster regular interspace short palindromic repeats, which is a source of small guide RNAs or a memory bank of past invaders of the bacteria. And then the CRISPR associated protein Cas9 is a cutting enzyme that basically cuts up the target mRNAs. Yeah, basically did talk about CRISPR earlier, so I'm not going to talk about anymore. And then small RNAs are used in the lab too. So if we have a cells, and then they glow cells with the protein that cause the cells to glow green or become fluorescent. But then when you put the cells in with siRNAs, the protein stops working and then the cells don't glow anymore. siRNAs are also used to vaccinate plants. Did I already go over this? I did, I just went over it. Okay. Oh, I went through translation also. Okay, I went through this 23. Genomics. Genomics. Oh my god, my computer is lagging. Okay, sorry, got distracted. Okay, genomics is a development and application of new mapping, sequencing, and computational procedures for the analysis of the entire genome of organisms. So, we have sequenced genomes, and yeah, and then there are a ton of them throughout the years. Wait, let's just get to the where I am. So 1978, the first virus genome sequence, but it wasn't counted because viruses aren't alive. And then 1995, first bacteria sequence as a circular genome started effort to start sequencing other genomes. Less than 2 million base pairs is very small. And then 1997, first eukaryotic yeast genome sequence, much larger than E. coli. New technology for genome sequencing was developed, 13 million base pairs. First, 1998, first animal genome sequence, the nematode C. elegans. It was 100 million base pairs. It made it big in the science world. And then 2000, fruit fly genome sequence, 120 million base pairs. It wasn't really known because in the same year, the first human genome sequence was discovered, but it was only a partial human genome. Or the first human genome was sequenced. It was only a partial human genome, but it created or led to a huge technological shift, and it was the start of genomics. And then from 2000 onwards, there was a rush to sequence other genomes, but then by 2010, the frenzy kind of died down. Okay. So to sequence a genome, a group of scientists get together and decide what they want to sequence. They write a proposal, they get approval, they get funds, they collect organisms and their DNA. They send the data to the sequencing center, and then you can obtain the data from the sequencing center and annotate it. And then you can publish your research. Yes. Okay. And the current state of genome sequencing. Here's all the sequenced genomes. Okay, and then the composition of the human genome, about 1.5% of the human genome is protein coding genes, and then about half is transposon based repeats, and then 44% is other sequences, and 4% is RNA genes and regulatory sequences. And then in the mitochondria genome, or most of it is protein coding genes. The other half is RNA genes, and then less than 2% is other sequences. Bioinformatics is a subfield of genomics. Is the application of techniques from computer science to problems from biology. A general problem that needs to be solved is that there's too much data to sequence, or too much sequencing data. So then the problems that can be solved are gene prediction and identification, where genes start and end, how many genes in a genome, gene and protein similarities, compare genes and proteins between species, gene and protein interactions, transcription factors and proteins, gene expression, what genes are expressed and when, protein structure, how do proteins fold and how the structure equals function, and evolution relationship between chromosomes, genomes, and organisms. Okay, and then next, the number of genes in the human genome is about 15,000 to 25,000 genes in the human genome. That was a surprising number to scientists, because how does the genome do what it does with only 20,000 genes? Well, we have alternative splicing that can explain that. So alternative splicing doesn't always occur in the same way in every tissue in every situation. Sometimes exons are left out. A single gene is able to produce 3 different mRNA, different proteins, and then have different functions. Sometimes exons are left in, not left out. Or left out, actually. They're actually taken out. Yeah, okay. So the number of genes across species is remarkably stable. Was not true for every species for eukaryotes, but almost all have around 20,000 genes or so. Parasites do not need a lot of genes, so they have less than that, so they use the genes of their host cell. Okay. So scientists found that some genes in there are some genes in common with eukaryotes. There are 458 core eukaryotic genes. Humans and mice have about 79% identity in amino acid sequences, and 65% of human genes are linked to disease, have a similar gene in fruit flies. So gene prediction is figuring out what parts of the gene codes for proteins have genes and which parts do not have genes. So you do that by sequencing the transcribed portion on the genome by sequencing the mRNA products through a lab technique called RNA-seq. RNA-seq is qualitative and quantitative. RNA-seq solves the issue of how to get mRNA out of cells. So to do that, mRNA has a poly A tail, then you put cells in a test tube and add like a broth for the cells to grow. You break the cells and then you take out the DNA in the solution into the solution, and then you add magnetic beads to isolate the RNA. The beads have tails full of diamines that interact with mRNA's poly A tails, and then hydrogen bonds are formed with the poly A tails and beads. And then you take a magnet and slide it at the side of the test tube to attract and pick up the beads. Then you wash the test tube of the rest of the stuff not attracted by the beads, and then you remove the magnet and apply heat. And then the test tube only contains mRNA. After you do all that, and then you stabilize the mRNAs since they're very fragile. And then you can find where the mRNAs come from, map it back to the genome, and identify where the genes are located. So to call a region of a genome a gene, several lines of information all have to converge. Similarly, oh, this could include similarity to known genes and evidence that is the region transcribed by RNA-seq. RNA-seq is sequencing DNA sequence that has the same sequence as RNA. Another lab technique is ChIP-seq. Genome browsers are websites designed to diagram genomic DNA, like UCSC Human Genome Browser or eEnsemble. You look at the human genome and see what is going on. When looking at genome browsers, you are looking at a snapshot of the genome. Okay, and then 24. So, what was the Human Genome Project? A U.S. government project coordinated by the Department of Energy and the National Institutes of Health. The goal from 1998 to 2003 was to identify the genes in human DNA, determine the sequences of the 3 billion bases that make up human DNA, store information found in the databases, develop tools for data analysis, address the ethical, legal, and social issues that come up from genome research. The first reference genome was to be a genome from several different people generated from around 20 primary samples taken from numerous anonymous donors across racial and ethnic groups. But the reality that was most participants were recruited from Buffalo, New York. 93% of the genome DNA was from 11 people. 70% came from a single African American male, and 30% was from a mix of about 20 people. The data was not a good representation of the population. Oh my God. So the Pangenome Project was an attempt to build better humans reference genome, a sequence of 350 people that is a much better representation of genetic diversity of humanity. The first step for Pangenome was accomplished by Chinese researchers who combined the genome of 58 people across China. So, how the human genome was sequenced, a first draft of the human genome was announced in the White House in the year 2000 as a result of a partnership between the public and private sectors. The public sector, or inside stories that the public sector universities were working on a large sequence of DNA trying to build a pangenome. They distributed work around since it was a large project. The private sector businesses decided that they were going to work faster than the public sector. Celera Genomics, a private company, was going to sequence and patent a large number of genes, and they were not restricted by the scope of their work. They were only interested in the genes or the profit. The public sector found out about that and they got scared, and then there was a race to prevent the patenting of genes and a race to patent genes between the two sectors. Eventually, they just worked together. So the technology we have for DNA sequencing cannot sequence large DNA. In any chromosome, we cannot sequence the entirety of it in one shot. Okay, and then We can sequence fragments of DNA by fragmenting a genome, breaking it into individual pieces, sequence the fragments, and reassemble them through overlapping parts into a single sequence. Once assembled, we can figure out how much of the genome encodes genes and how much of the genome encodes other parts like non-genes. The ENCODE project, or Encyclopedia of DNA Elements, aims to identify and describe parts of the human genome that are functional. The main outcome of the project has been to show that a significant portion of the human genome is transcribed and used for something. The ENCODE project is an ongoing project that has expanded to other genomes, such as mice and Drosophila. Eventually, we can go to a genome and find out when and where it might be expressed, like only in adults or only at night. But that requires you take the genome and sequence it under different conditions and developmental stages and in different tissues. The ENCODE project was a large project that is restricted to a few genomes. So there needs to be a lot of interest in a specific genome for a project to be done on the genome. So there are issues with that, with like studying genome, which is like who the human genome belongs to. So patents can lead to innovation and discovery, but patents are temporary, like 20 years. But in a fast-changing world of medicine, a 20-year patent is a long time, and our genes are our own, so we shouldn't patent them. Example was the BRCA gene, which was patented by the private sector. The BRCA analysis test prices went up with patenting, and the question of why we should pay to find out the information of our own bodies came up. The BRCA is closely related to breast cancer. If an individual has an allele on the gene, there is a high chance of breast cancer. Right. Okay. So when it was patented, it was less accessible to a lot of people. So in 2012, the Supreme Court ruled that naturally occurring sequences cannot be patented, but modifications to the sequence of the human genome can. Okay. Okay, what we learned from sequencing the human genome? We learned some remarkable things about our ancestry, like how Christopher Lee found out he was related to Charles I, or how any white person with white skin is related to Charles I. And yeah, and then African Americans are more diverse than any other groups of humans. And then pedigree collapse is a phenomenon that explains how every individual on Earth once came from a single ancestor. If you go back a few generations, there are more ancestors than there ever were on Earth, and you will find that some ancestors were even related to each other and inbreeding happened between them. In a pedigree collapse, only a small group of people are contributing to the diversity. Large families pass on diversity, but we are a lot less diverse than we think we are. Another thing is that at some point during evolution, there were different groups of humans, like Homo sapiens or Neanderthals. Different groups coexisted in Africa. When ancestors of Homo sapiens started moving out, they came into lands occupied by Neanderthals. The thought was that our ancestors kicked out the Neanderthals by waging war and displaced them, and then they died out. But by sequencing the human genome and Neanderthal DNA, we found out that people with But that didn't happen because our ancestors didn't kick out the Neanderthals and displace them, because through sequencing human genome and Neanderthal DNA, we found out that people with non-African ancestry have between 1.5 to 2.1% Neanderthal DNA. That suggests that our ancestors bred and got rid of the Neanderthals after. We can recreate a tree of how humans have evolved by we can create this tree by comparing sequences of DNA in different areas to the human genome. In 2010, Denisova hominin was described from a single finger bone found in Papua New Guinea. People in Papua New Guinea have particularly long stretches of Denisovan DNA. Neanderthal DNA was sequenced, and then we found some similarities. It was a likely evidence of interbreeding between the two groups of humans. So how we compare the human genome? We actually have more than one human genome, paternal and maternal lineages. Paternal lineages is the Y chromosome, and maternal lineages is the mitochondria DNA or mitochondria. We can recreate history based on only paternal lineages or only maternal lineages, since those are independent of each other. So if they tell us the same thing, then the information about history is correct. History that shows that all of humanity starts off in Africa, then they leave to Asia, then to Europe, and then finally the Americas, and explains why diversity in Africa was so high. Early on, there was a huge migration to Papua New Guinea in Australia, probably before Europe was colonized. Okay, this is the last slide I'm going to talk about. Okay, blue eye color is often due to a mutation on chromosome 15 that prevents production of melanin. So people with blue eyes lack pigments. And then we have mutations at different levels. We have base level mutations, or point mutations. The types are transition and transversion. Transition is when you go from pairing to purine or pyrimidine to pyrimidine. And then transversion is when you go from purine to pyrimidine, and it has a bigger side effect since there's a bigger change. But for transition, there's no change in, or for both, there's no change in the number of nucleotides, but there are more transitions than transversions. Okay, let's just repeat. So, next is insertions and deletions or frameshift mutations. Adding nucleotides and removing nucleotides cause chaos in how nucleotides are translated or read. It shifts the reading frame. Inserting 3 nucleotides, though, is not a lot of issue since the strand of nucleotides is eventually read correctly. Muscular dystrophy encompasses a group of genetic disorders that result in the wasting of muscle tissue. People cannot control muscles and have tremors as a side effect or other side effects. Muscles contract by anchoring to actin, or muscles to exert a force. Actin, skeleton of the cell, anchor to cell walls. Muscles anchor onto actin to exert a force on the actin that is staying still. Actin are muscle fibers. Dystrophin connects actin and muscle anchor. individuals have a faulty dystrophin molecule if they have muscular dystrophy. There are two forms. There's Duchenne, which is that forms during early childhood and makes someone weak. And then Becker with symptoms later in life, and it's not as severe as Duchenne. Dystrophin protein is involved in the structure of a large complex in muscular membranes. When missing or non-functional, the entire complex is affected and it leads to dying muscle tissue. Deletion that is large or small. Muscle wasting occurs when ability to regenerate muscle is exhausted. So Becker, the mild version, is a large deletion, but the reading frame is still intact. And then Duchenne is a severe condition and is a small deletion, but the entire reading frame is changed. Okay, repeat expansion. Certain sections of our genome are repeated. Trinucleotide repeat disorder. Cause of disease is traced to having too many copies of a certain nucleotide triplet in the DNA, like CAG. Trinucleotide repeats repeat themselves in protein. The number of repeats affects how protein folds. So like Huntington's disease. Huntington's disease is the repeat of CAG. It's neurodegenerative, brains shrink, and individuals have dementia. It's autosomal dominant. One copy of the repeats have allele for the disease. Or if you have one copy of the repeats, then you have the allele for the disease. Huntington's disease exhibits symptoms later on in life. Yeah. There's no cure currently for this disease. We have protein level mutations. Silent mutations, no amino acid change. It's neutral conservative, missense. Amino acid changes, non-conservative, nonsense, stop codon, premature, end or stop. Frame shift, insertion or deletion of amino acid. So for silent, the letters change meaning and the meaning of the but the meaning of the sentence doesn't change. So if you have the dog saw the fat cat, then the new sentence is the dog saw the fat cat, and then the cat has a K instead of a C. So you changes one codon for an amino acid to another codon for an amino acid, but there's no change in amino acid. Missense, the dog saw the fat cat, but then you change it to the dog ate the fat cat. It changes the codon for one amino acid to another amino acid, and it may alter protein function. Missense, you have missense, you have sickle cell disease. So the original sequence of the normal red blood cells have an amino acid for glutamic acid. But the new sequence for sickle cell red blood cells change glutamic acid to valine instead. And valine, okay. Then valine has no electrical charge, it's neutral and it's hydrophobic, while glutamic acid is hydrophilic and has a negative charge. Glutamic acid and valine have different properties, so there's going to be different effects. And then for nonsense, the mutation is just like the sentence just cuts off. And it changes the codon for amino acid into a stop codon. And it may lead to a short and non-functional protein. So for nonsense, we have beta thalassemia, which is a hereditary blood disease linked to severe anemia, growth delays, and blood deformities. Occurs when transcription of beta globin gene that codes for part of the hemoglobin protein is prematurely stopped due to a stop codon. And then we have frameshift mutations. When deletions and insertions change reading frame, and it results in a non-functional protein. And this is Tay-Sachs disease. Autosomal recessive disease causes the dying of nerve cells, and 4 extra nucleotides are inserted into the hexa gene and cause a frameshift, leading to the production of a non-functional enzyme, hexasaminidase. Yeah, this is Tay-Sachs. Next, we have chromosome level mutations. So 23 chromosomes went through many changes throughout history. Deletions are missing segments, insertions is extra chromosome segments, duplication is segment with extra copy, inversion is same sequences of nucleotides, but they are flipped, and translocation is 2 chromosomes exchange segments. Reciprocal translocation, or a segment of chromosomes detach and each segment attaches itself to the other chromosome. So this is Parkinson's disease. It's a rare chromosomal disorder that is often due to duplication of SNCA gene. Individuals can't control their muscles because signals don't always get through. SNCA gene encodes alpha-synuclein, and it involved in the physiology of neuronal synaptic vesicles. Duplication leads to the death of neurons that produce dopamine, and it leads to the disorder because there's lower levels of dopamine. Neurons transmit messages, electrical signals. When 2 neurons come together, there are no electrical signals being sent. Electrical signals is encoded into a chemical signal and is diffused into the next neuron, which takes the signal, translates it, and sends it back up. Dopamine carries these signals from one neuron to the next neuron. Translocations. So we have VLC lymphoma, which is a blood disease, and it's a translocation from CR18 to, or chromosome 18 to chromosome 14. The promoter for antibody is placed in front of the BL2 gene, or the BL2 gene is placed in front of the promoter for the antibody, and antibodies have strong promoters, so that means a lot of BLC2 genes are made, and then BLC2 genes are, or BLC2, or the BL2, BLC, or BCL, well, I've been saying it wrong. Okay, the BCL2 gene is an oncogene, and if activated, causes cancer, so it's usually silenced. But in this case of BCL2 lymphoma, it's activated and it creates a lot of them. A lot of BCL2 genes are created. Okay, aneuploidy is the abnormal number of chromosomes. So 2n+1 is trisomy, or Down syndrome. 2n-1 is monosomy, or Turner syndrome. And abnormal number of chromosome sets is monoploid, triploid, and tetra. Trisomy 21 is non-destruction during meiosis 1 or 2. Non-destruction is usually deadly, but sometimes you can survive from that. Non-destruction of chromosome 21 in human lead to a trisomic nucleus or Down syndrome. You'll accumulate many this Down syndrome. You'll get a lot of mutations in your lifetime, like somatic mosaicism, and then there's a condition called somatic mosaicism, which is not all your cells have the same genotype. A 2024 study showed that 1 in 40 bone marrow cells showed massive chromosome alterations, extra chromosomes, and big rearrangements. As you age, the number of cells with changes get bigger, likely contributing to age-related diseases. The last is AlphaMissense. It was announced in September 2003. It was an AI-based program that tries to predict what phenotypic effect of multiple mutations in the human genome would be. Okay, that's it. I talked about everything else.